So hello, my name is Youngmo Kim and bioanalytical chemist at Pacific Northwest National Laboratory. Thanks for having me here today to present the multiomics capability what we have. And as you probably know, or maybe not, but ABF test team at PNNL has been a part of ABF since the consortium started. So I will quickly go through our capability within 10 minutes. Next slide, please. So everyone here knows that microbial system is somewhat an efficient and promising tool to produce some chemical compounds, can replace the products from petroleum industries, but still, many bacteria or fungal systems are not easy to understand in terms of their functions over the information what we know about their gene. So even biomanufacturing, we do some modifications, lots of add in or add out. So it's still hard to understand what really happening inside of a cell. So even a long time ago, we got all the gene informations from a lot of microorganisms, but we still have limited information of their functions. So I don't want to go over all the slide detail, but yeah, so that's what we want to pursue to in the future. Next slide, please. So we can characterize microbial system by many different advanced analytical tools. And as you can see here in the central dogma, yeah, we see lots of DNA or messenger RNA or proteins and or metabolites or lipids and everything. So we want to capture, we want to know how they change over under different condition or gene modification. So we can measure them by many different types of analytical instruments, but what we have is mass spectrometry and lots of different chromatography. So we can measure their proteome and metabolome and some modification of the glycan or lipids. And also with the help with the computational modeling, we can calculate the metabolic flux. So we can capture how the carbon goes from the substrate to final products. So next slide, please. So before digging into the deep biological system analysis, we want to see how they look like first. So we have an electron microscopic capability to capture them. So why do we need to take these pictures? Because our measurement begins with extraction. So we test the many different types of extraction methods to disrupt the cell wall or cell membrane and structure. So these four are representative host strains what we use. So the left one is gram-positive Bacillus subtilis, and we see their rod shape. And the second picture is Pseudomonas putida, and we see the scale bar. So we see that the Bacillus is longer than putida, and even we can see some EPS, extracellular polymeric substances, usually secreted by the bacteria, and then they can make a biofilm. So even we can see their presence or absence by the electron microscope. And the right two pictures from the eukaryote fungal system. So the third one is the unicellular fungi, the Yaroia lipolytica, and the last one is the Aspergillus niger. It's very long and very hard shape. We had a lot of photos on this by electron microscope after the modification or extraction disruption by using bead beads or sonication or some enzymatic lysis of the cell wall. So I think that is important to understand their unique phenotype difference at the beginning. Next slide, please. So there are many different options to choose for the extraction method, but we haven't improved our extraction method. We call metabolite protein lipid extraction. abbreviated as an amplex. So during this extraction, we can separate polar metabolites and the top layer. I'm saying about the photo on the left, and we can see denatured protein disk in the middle, and at the bottom we will get non-polar metabolites, sterol. For fungi, we see a lot of ergosterol. It's a sterol type of metabolites present in the cell wall, and also other intact lipids. So once they were extracted from our extraction, we can do many different types of analysis. So bottom one is we can see discovery proteomics. So that is just global proteomics. We can measure all the proteins as possible as I can. So from our previous collaboration with many different industry partners or the other national lab, we typically measure about 3,000 each of proteins from bacterial system, and fungal system, we were able to detect more proteins, about 4,000 or even 5,000 proteins. And that's for discovery types of a measurement. And at the same time, we also measure certain protein and peptides in the system because we do lots of gene modification and editing. So our collaborator had an interest how they actually expressed or knocked out. So we were able to measure them by specific targeted proteomics analysis. Not general, but and also we can do post-translational modification proteomics. So in general, phosphorylation or acetylation and ubiquitinations, and then lots of things can happen on proteins. So we can also screen those PTM information from the protein side. And the right side, and then extracted the metabolites and lipids can be analyzed by mass spec, by GCMS and HCMS, and with the help of computational modeling, and we can calculate the metabolic flux analysis. Next slide, please. So we have these two types of, in general, kind of proteomics. So upper one is targeted. So we need to add heavy isotope labeled substrate, and then we can measure both of them to measure their intensity. So very focused analysis. And the bottom one is we can just do the full search with all the proteins. So as we wrote down at the bottom, so we have a lot of CRADA project, and then we really helped our collaborator, and we guided them to measure overexpressed enzymes and proteins, or during their promoter improvement, we were able to see their up and downs. The collaborator told us that it's really helpful to get further gene modification and engineering. Next slide, please. Oh yeah, yeah. The next slide. Then that is our improvements. Nowadays, all the chromatographic technology and mass spectrometry sensitivity has been increased a lot. So previously we used the data-dependent acquisition is more traditional way, but now we can do data-independent acquisition. So we can increase more coverage, and also we lowering missing values, and we also can reduce the all separation time. So we still keep improving this capability to capture more. Next slide, please. So in Metabolomics pipeline, it's simple. We just collect cells or culture medium, and then we can separate them, and we can extract metabolites from the cell biomass. At the same time, we can just measure the metabolites in the medium, because sometimes unwanted byproducts are present in the medium. That's not what we want. So we can easily screen it by mass spectrometry. Next slide, please. So GCMS side, we can get good separation by over time. So top chromatogram is separation by time, and the bottom one is the mass spectrum at given point. So small metabolite it looks early, and bigger metabolite is late. And then once we get spectrum, then we can search that information to our database. Or click, please. And like this is a citric acid mass spectrum, and we can accurately identify and quantify their presence, absence, or their amount by mass spectrometry. And PNNL has been working more than 15 years to improve the accuracy and the data generation consistency. So, and also at the same time, we have trying to get, because omics, metabolomics is based on the size of a database. So we are trying our best to improve our whole the database size. Next slide, please. And this is a LCMS side. So we can do targeted or global metabolomics using LCMS. And previously with the CRADA project with Agile technology located in Santa Clara in California, we developed a very fast and efficient screening tool to capture highly preferred 64 metabolites in our host system. And we published a paper. It was a really fruitful collaboration with the industry partner for this, and we are working on other future things as well. Next slide, please. And this is a global HMS. So global HMS, we will get all the peak informations, and then we align them, and then we use that information, the accurate the MOG values and their fragmented spectrum to other database, and then we can report as much as you can. Because GCMS and LCMS, their coverage are a little bit different. So LCMS can provide the bigger molecules or vitamins or other things. So it will be good addition to the strain characterization. Next slide, please. So the last thing is the fluxomics analysis. We can use the isotope enriched substrate to monitor their metabolites. So we can measure the addition of the incorporation of isotopes, and we can use the information to calculate the flux. So right one is the example of our recent publication. So we use Pseudomonas putida, and we try to change their metabolic pathway to produce a dye molecule, and we were able to generate this pathway map. So next slide, please. So this is summary. So proteomics and metabolomics and metabolic flux analysis can provide some valuable information inside of cell, what is happening. And in addition, well, we have a ICP-MS for meta-analysis. And as I shown before, we have electron microscope capability to capture their morphology. So CRADA collaborations with the AFF test team is highly valuable and efficient for gaining better understanding of industry partners' biology system. So that's why I present here. Thank you.